reaching up into the water like the branches of trees. They are thousands of organisms living together as one, and together they provide shelter and protection for many more.
This is really important for a lot of different marine organisms and provides a refuge for these organisms.
Once a common sight, elkhorn and staghorn corals were the dominant shallow reef-building corals in the Caribbean and Florida.
Coral reefs are under extreme stress today from a variety of causes, both globally and locally.
In the past 30 years, elkhorn and the staghorn have declined dramatically, between 90 and 98% throughout the Caribbean. They're now listed as threatened under the U.S. Endangered Species Act.
And are much reduced from their former glory.
Primarily, disease, as well as some other factors, has led to the drop in numbers of elkhorn and staghorn colonies. But while these corals have declined in recent years, their hybrid appears to be increasing in parts of the region.
What's really cool about this hybridizing system is that the hybrid is not found in the fossil record. They seem to be increasing recently, likely due to the decrease in the parental species.
Often found in really shallow water, this hybrid, commonly called fused staghorn, closely resembles its parents.
You have some that look very much like staghorn and then others that look like elkhorn. Most of the ones that I've seen throughout all my study sites in the Caribbean have more of a staghorn look to them, but they're more tightly clumped than the branching staghorn.
Could these hybrids be better equipped at dealing with environmental stressors? Might they be filling a niche left behind by the decline of their parents?
Major funding for this program was provided by the Batchelor Foundation, encouraging people to preserve and protect America's underwater resources, and by Divers Direct, inspiring the pursuit of tropical adventure scuba diving.
Dr. Nicole Fogarty is an assistant professor at Nova Southeastern University's Oceanographic Center. She studies the hybrids at various sites across the Caribbean.
I was really interested in this hybridization between these two threatened species. There's very little that's known about the hybridization.
Since 2005, she's been making regular research trips to a small island off the coast of Belize.
The island is out on the Mesoamerican Barrier Reef, so you really can literally roll off the island and be on a coral reef. There's just a very nice reef very, very close to the island.
The Mesoamerican Reef stretches along the coasts of Mexico, Belize, Guatemala, and Honduras. It is considered the largest barrier reef in the Western Hemisphere, covering nearly 700 miles from the northern tip of the Yucatan Peninsula down to the Honduran Bay Islands. Caribo Key is located 15 miles offshore from the Belizean town of Dangriga. It has been a research facility of the Smithsonian Institution since 1972.
We just celebrated our 40th year anniversary, And we've got over 930 scientific publications that have come from the island, including a few books that have all resulted from the work at Cariboat. It's really contributed a lot to our understanding of coral reef ecosystems in the Caribbean.
The island is an ideal place to study the hybrid because of its proximity to all three types of corals.
There's a wonderful population just off the southern end of the island. It has both the staghorn, the elkhorn, and the hybrid of those two species.
When Nicole first started coming to Cariboat Key, she wanted to understand why the hybrids might be increasing in numbers.
One possibility is that there's an actual increase in hybrid formation. So the egg and sperm of staghorn and elkhorn are mixing to form an embryo that's a hybrid that will eventually go and settle and grow into an adult hybrid coral. The other possibility is that we have an increase in asexual fragmentation. These corals are branching corals, and so they can break off and fragment, reattach to the bottom, and then continue to grow. So that's a very important aspect of the life history stage. So you could imagine if there's just a few rare hybrid events that formed decades ago, these hybrids could just fragment repeatedly over the years. The other possibility is that the hybrid is actually more viable than the parental species. It might show increased fitness. It's called hybrid vigor. And so we have hybrids that have been established over the years, and when you have the disease outbreak and disturbance and other things that have led to the decline in the parental species, the hybrid is actually able to withstand all those different events, and that's why they're increasing in numbers and are higher in abundance at some of the sites.
To test these hypotheses, Nicole decided to conduct a number of experiments when the corals were spawning.
Staghorn and elkhorn coral only spawn once a year, usually in late summer, July, August, or September. So that means one night out of the year, they release gamete bundles, and those gamete bundles are full of both egg and sperm. Each individual polyp that makes up a coral colony will form one gamete bundle. It takes the polyp about 9 months to create the egg and about 6 months to create the sperm. So there's a lot of energy being invested in forming these gametes. The gamete bundles are released in one synchronized pulse. They float up to the surface, break apart, mix with other gametes from other individual corals of the same species, or sometimes hybridize, and then fertilization occurs at the surface. The resulting embryos then float around for a couple days before developing into larvae, and then the larvae will float around for a few more days before they start swimming down to the bottom, looking for a good place to settle. They'll attach to the bottom, metamorphose, and grow into an adult coral.
Staghorn and elkhorn corals are very plant-like creatures.
And so really when you're thinking about corals, even though they're animals, their evolutionary background and evolutionary trajectory are much more like plants. They are hermaphrodites. They're attached to the bottom, just like adult plants are. They broadcast spawn their gametes, like many plants do. They're long-lived, like plants. And they also can asexually fragment. They asexually propagate, like many plants.
Using a tent-like net, Nicole collected the gametes of both elkhorn and staghorn corals on the nights they were spawning.
We bring those gametes back into the lab. We can separate the gametes out into sperm and stock solution, and then we can play mad scientist where we do all kinds of different fertilization assays. Now that we've separated them out into our sperm and egg solutions, we're going to conduct a series of fertilization experiments. And so we're looking at fertilization between the elkhorn and the staghorn, and this is crosses between the staghorn eggs and the elkhorn sperm. And now I'm going to add 1 milliliter of eggs to the vials. After I complete this cross, then we'll do the reciprocal cross and mix staghorn sperm with the elkhorn eggs. And then we can do it with sperm competition. That's when we mix both the staghorn and the elkhorn sperm together and then introduce the sperm to the staghorn eggs or introduce the sperm mixtures to the elkhorn eggs. And that's more likely what occurs during coral spawning because these species spawn at the same time. And so the sperm are going to be mixing, competing for who's going to actually fertilize the eggs. Once all the crosses are done, we'll let them sit for 3 hours and then we'll score fertilization. When we score fertilization, what we're looking for is the number of unfertilized eggs and the number of dividing embryos.
With these fertilization experiments, Nicole tried to determine if there may be gametic incapability, which means one species' sperm can't fertilize the other species' eggs.
What we found out is that both the staghorn eggs and the elkhorn eggs are able to hybridize, but the elkhorn eggs are a little more resistant to hybridization than the staghorn eggs. The staghorn eggs can be fertilized by the elkhorn sperm just as easy as their own species sperm. In the sperm competition assays, we found a similar result. The eggs of elkhorn are a little more resistant, and then the eggs of staghorn are more easily fertilized by not only their own species sperm, but the elkhorn sperm as well. So it can happen in both directions, but hybridization occurs more frequently with the staghorn eggs.
Next, Nicole looked into the viability of the hybrids.
The hybrid might be sterile, such as the mule example when a donkey and a horse mate, or it might just be inviable. So it has decreased fitness and it's not able to persist in the environment. Perhaps it's more susceptible to disease or temperature fluctuations. So these are the type of things I wanted to look at. And what we found is that the hybrid was not inferior across any life history stage that we looked at. That include the larval stage, during settlement, throughout metamorphosis, and also as an adult. We also found that hybrids can persist in the shallow marginal environments they're often found, but also can persist and grow in the perennial species habitat without a problem.
And Nicole made another interesting discovery at her study site in Belize.
The hybrid, even though they're often in just 2 or 3 feet of water, are no more likely to bleach than the perennial species that are in deeper water. And so this could be that the hybrid's more tolerant of fluctuations in temperature. Not only increase in temperature during the summertime, but those shallow areas also cool off really quickly during the wintertime. And there's also a lot of UV irradiance blasting those corals in that really shallow, clear water. So that's something I'm going to be exploring further.
Coral bleaching occurs when the animals are very stressed. There are a variety of stressors that can cause bleaching, including exposure to extreme temperatures. Extreme temperatures are likely going to increase in frequency in the future because of climate change.
Coral bleaching is when the corals lose their symbiotic dinoflagellate that's in the tissues of the coral. So corals have a symbiotic relationship with a dinoflagellate we call zooxanthellae. And the zooxanthellae is in the coral tissue. It gives the coral the golden or brown colors that you see. And what occurs is when the coral gets stressed out, that symbiotic relationship is going to break down. And so they lose that zooxanthellae, and then what you see is the white coral skeleton through the translucent tissues. Now, the dinoflagellate, the zooxanthellae, is important for the corals because it gives the corals a lot of its nutrition. Corals can capture small zooplankton in their tentacles, but most of the nutrition that the coral gets is through the zooxanthellae. So when this symbiotic relationship breaks down, the coral is going to have decreased growth rates, it's going to have reduced reproductive capabilities, and so it's going to really negatively affect the coral.
One thing that is still unknown at this point is whether or not the hybrids can mate with each other.
We know that hybrids aren't sterile because from the molecular signature, we know that the staghorn and the hybrid can mate.
However, to date, no second generation of hybrids has been discovered.
There's a couple different hypotheses of why we might not see a second generation. One of which is that the sampling effort was pretty low. It was only at 3 geographic sites and pretty low sample size. So perhaps second generation hybrids are out there, but it'll take further sampling. And that's something I'm looking at a part of my research at Nova Southeastern University. The other possibility is that if hybridization is increasing, as I believe it is, then we might have more generations in the decades to come. It's just such a new event that they're increasing in numbers that they haven't had an opportunity to create a second generation. What's really interesting about this system is that the perennial species are found in the fossil record for millions of years, but the hybrid has no fossil record, so it's a relatively recent event. And there is evidence that it is increasing in some sites. There are sites in Belize and in Curacao in Florida where we know the hybrid was not that site just 5 years ago, and now we see hybrids there. We also know from genetic studies that the hybrid populations are composed of multiple genotypes, meaning that there's separate hybridization events that have occurred and not just one rare hybridization event that has asexually fragmented. What I think is currently going on in this system is that with the decline of the perennial species over the past 30 years, we actually have an increase in hybridization. And how that would work is that if you imagine back before the 1980s, you had really extensive thickets of both staghorn and elkhorn. They were the primary reef builder in the shallow reef environments. And so when staghorn or elkhorn spawned, the eggs are immediately going to be swamped by their own species sperm. And occasionally you might have had hybridization if a couple eggs went unfertilized, but it was a pretty rare event. Well, now you have these populations of elkhorn and staghorn that are really sparse. And so if you imagine when they spawn, the eggs are going to go unfertilized. And at least with the staghorn eggs, whatever sperm they run into, whether it's their own species sperm or an elkhorn sperm, that's going to fertilize it. So we have this increase in hybrid formation because the perennial species have decreased in numbers.
Long-term monitoring is necessary to truly understand the changes occurring on the reef.
Only through long-term observations over time can you evaluate and find new discoveries, and I think it's paid off in a big way.
In 2011, Nicole created a demographic study off the south end of the island.
As far as we know, we're the only demographic study Caribbean-wide that looks at both the elkhorn, staghorn, and the hybrid. We established 7 circular plots, and these are permanent plots. So every 3 to 4 months, we go back and look at every individual elkhorn, staghorn, or hybrid in these plots.
Each of the 7 plots has a buoy at its center.
And then we took a transect line and extended it 7 meters from the center. We then tagged every single coral colony within that 7-meter radius in that circular plot. We also mapped it, and how we do that is we take the transect line from that center buoy and extend it out to the focal colony, and then someone in the center buoy has a compass looking at their compass and then will give us the different numbers for that compass bearing. Whoever is at the focal colony will then write down the distance from the center buoy and then also that compass bearing that the person at the center then gave them. That way we can mark each individual coral colony and where it is.
Next, tissue samples were taken for genetic analysis.
We take small tissue samples, about a centimeter, just the very end of your pinky. It's only about 1 month of growth for these corals, so it doesn't hurt the corals at all. We take that tissue sample and preserve it. Then we take it back to the laboratory and we use microsatellite markers to be able to genotype. And then that gives us an idea of how many separate genotypes are in our population and how many of the different colonies are actually from asexual fragments. So far we have nearly 600 corals tagged and followed over that period of time. The elkhorn coral out there is really genotypically diverse. We have over 30 genotypes out there. The staghorn is less genotypically diverse, and there's only a couple unique genotypes, and the hybrid has about 4 or 5 unique genotypes.
Nicole based her demographic study on a protocol designed and used by other scientists in the Caribbean.
We can compare our results in the Western Caribbean with their results in Florida and elsewhere in the Eastern Caribbean. Every 3 to 4 months we come back and we photograph each individual colony. We also assess each individual colony, and so see if there's any damselfish bites, see if there's any predation, coral bleaching, disease, etc.
Nicole also takes length, width, and height measurements of each individual colony, and then estimates the percentage of live coral for each colony.
We're also interested in how quickly these corals grow. So what we do is we take a beaded cable tie and put it about 2 centimeters away from the tip of the coral. The coral grows from the end, and so it will continue to grow and extend, so we can measure from that beaded cable tie to the very end of the coral and see how much it grows every time period that we come out here and sample. We're going to be doing a lot of spatial analyses and being able to look how disease spreads across this population and coral bleaching, predation, and see how that changes year after year and how it affects different colonies and may spread to neighboring colonies.
In the few short years that this project has been ongoing, Nicole has already made several interesting discoveries.
We established this project just a few months before a major disease outbreak. The other aspect is that we've already lost individual genotypes from that disease event. And lastly, we're seeing some new recruits, at least colonies that we think are new recruits. Once they get a little bit larger, we'll be able to take a tissue sample and genotype them and verify that it's a unique genotype compared to the rest of the individual colonies out there.
So it's really a valuable data set. There's nothing else quite like it in the Western Caribbean. There's some similar monitoring going on in the Florida Keys and also in the Virgin Islands, but this will add a new data set for a region, the Mesoamerican Barrier Reef region, which is really much less explored.
What we ultimately would like to do is put this data into population models. And so then we can determine whether this particular population is stable, increasing, or decreasing.
There are still many questions left unanswered that Nicole hopes to study.
Because the elkhorn and the staghorn corals are the only current threatened species in the Caribbean, it's really important that we have a better handle of what's going on with them, both from an ecological standpoint and also an evolutionary standpoint. And so this information not only will provide some data needed to understand the evolutionary trajectory of these species, but also what's going on from the ecological perspective. If the hybrid is more tolerant to increased temperature or UV irradiance, then it's going to be able to withstand some of these harsh environmental changes that we may see associated with global climate change. If we lose one or both of the parental species, it'll be interesting to know if the hybrid can reproduce among itself and can provide a genetic reservoir for some of these genes that we've lost through the extinction of the parental species.
It's an avenue that's worthy of research and may provide some great information on how coral reefs can be restored and thrive in the future.
Major funding for this program was provided by the Batchelor Foundation, encouraging people to preserve and protect America's underwater resources, and by Divers Direct, inspiring the pursuit of tropical adventure scuba diving.